All right, guys, chapter 15 is a fairly short chapter. There's not much. There's a few diseases, a few things that about chromosomes we need to talk about. Remember, Mendel did not know anything about chromosomes, okay? And of course, I don't know if I should, we can look it up. Mitosis, when the very first people saw these little thread-looking things on the microscope, they called it mitosis. They had no clue what it was, chromosomes. So they called them mitosis, and mitosis in Greek means thread. Okay. Anyhow, so locating genes along chromosomes, Mendel's hereditary factors were purely abstract when first proposed. Today we can show that the factors genes are located on chromosomes. That's right. I talked about it. This is a chromosome. This segment is a gene. That segment is another gene. That segment is another gene. That segment is another gene. And that's what they are. And then, of course, you have two chromosomes. So they are one allele, right? So the other one, two alleles. The location of a particular gene can be seen by tagging isolated chromosomes with fluorescent dye. Highlight the genes, yes, right, just like here, they're showing it to you guys here. Okay, so back to Mendel, what happened, that yellow rounded seed, and then he crossed them together. The first generation were all yellow, and then, as you know, in meiosis, we already talked about that before, the chromosomes segregated, they went to, these are the different possibilities. And then what happened, then when the gametes were formed, look, you have, in this case, you have two different types of gametes. In this case, you have, again, two different types of gametes. When I say two different types quickly, this is one type, right? This is the other type. They both are similar. Same as here, okay? So the long blue and long red, and long blue and long red, here is long blue and short red, and long blue and short red. So these are different possibilities. Law of segregation, and he's explaining them for you right here perfectly. Law of segregation of independent assadi. For his work, Morgan's choice of experimental organism, he used a Drosophila, which is a fruit fly, and then they're excellent organisms to study for genetics. They multiply fast, and they only have 8 genes, somewhere near 8 chromosomes, and they're excellent specimen to study. And they only have 4 pairs here, 4 pairs of chromosomes. It means 8 chromosomes. 4 pairs, that means 8 chromosomes. And then Morgan noted wild type, the ones that is found in the wild, they call them wild type, and you will see that. Or normal phenotype that were common in fruit fly populations. Traits alternative to the wild types are called mutant phenotype. For example, right here, he is, he is calling the red eye right here, that would be a wild type, if I'm not making a mistake. And this one would be a mutant type. It means these are more common in nature. So he called them wild type. He didn't call them normal or abnormal or dominant, recessive. He stayed away from them. So here they are. If in one experiment, Morgan mated male flies with white flies, mutant, okay, the male, the white ones are mutant one, with the female flies with a red eye, which is the wild type, okay, female and male, and he got all red, okay. The F1 generations all had red eyes, male and female, both of them, okay. So in the F2 generations, he showed a 3-to-1 ratio of white eyes, but only males had white eyes, okay. So he said, what is going on? Why only male have the white eyes? Okay. Morgan determined that the white eyes dominant alleles to be located on the X chromosomes, the sex chromosomes. And right here, it's talking about that, the sex chromosomes and what is happening. I'll let you study that. Here is the X and Y chromosome. They are, you know, in animal kingdom, we human use X and Y, okay, you guys? In animal kingdom, they can use different genotype. They are using different genotype, such as ZW and ZZ, or in the case of bees. Study that on your own. I'm not too crazy about it too much. Short segments on the end of the Y chromosomes are homologous with the X, and allowing the two behave like homologous during meiosis in male, and a gene on the Y chromosomes called SRY, very important, sex determining regions of the Y chromosomes responsible for the development of testes and the male reproductive tract, pretty much in the embryo. So the presence of SRYG on the Y chromosome makes a male and female. Females are XX. They do not have the Y. They do not have this gene. So they develop the female reproductive tract. A gene that is located on either sex chromosome is called a sex linkage. And then sex linked gene, which as I promised you, we're going to talk about colorblindness. Genes are Y chromosomes are called Y-linked genes, and there are a few of these genes. Genes on the X chromosomes are called X-linked genes. Oh, sorry, I clicked too much, did I? Here we go. No, yeah, I did. I think I did. Yeah. Okay, X-linkage. Genes follow specific patterns of inheritance. For example, for recessive X-linked trait to be expressed, a female needs 2 copies of the allele homozygous, a male needs 1 copy of the allele hemizygous, and X-linked recessive disorders are more common in female than. Okay, right here. So the way your textbook is designating them by N, and the way I like to designate it by Y, by different, let's see, I think that's it. So let me write down the way I like to say it. So if the female, let's say 2 parents, 2 person get married, and one of the female, she has a carrier, and the male is not a colorblind. So they get married, and they have, of course, you form the Punnett square on them, right? They have a daughter that is colorblind. They have a carrier, I'm sorry, not colorblind, sorry about that. That is a carrier. They have another daughter, which is normal. They have a son, right? They have a son that is colorblind. And they have another son, which is perfectly normal. Okay, so it is possible. This is a carrier, this is a carrier, this is a normal, this is colorblind, and this is a normal boy. Let's say the colorblind boy goes ask a girl that, are you colorblind or not? The girl says, no, I'm not colorblind, I'm perfectly fine. The boy has been colorblind, and he says, hey, listen, I don't want to get married to anybody colorblind. I don't want to have any child that is colorblind. I love my children, my future children. I don't want them to be colorblind. And the girl says, hey, listen, I'm perfectly normal. You pick up a color, I will tell you what they are. Red is red, and black, of course, they can see that. Blue, green, they all can see that. Okay, they get married. Here are the possibilities of the sperm. And here are the possibilities of the egg. So they have a child, a baby girl, and guess what? Baby girl is colorblind. Baby girl, and he says, oh my God, what happened? I thought you're okay. Say, well, I'm sorry, I didn't know I'm carrier. They have a boy, he's colorblind. The guy gets really mad. And they have a third child. The third child is going to be a carrier, but the daughter can see normally. They have another boy, and the boy is also normal. Okay, so normal, male, carrier, female, colorblind, male, and colorblind, female. Colorblind, female. So, some disorders caused by recessive alleles on X chromosomes in humans, colorblindness, mostly X-linked, yes, dunting, muscular dystrophy, hemophilia, they're all, those are some of the other disorders are sex linkage. Unlike genes, recombination of unlike genes, independent assortment chromosomes, offsprings with phenotype matching one of the parental phenotype are called parental, yes, and the other one is called recombinant. So let's look at it. So in case, if you're looking at this, these are parental and these are recombinant because they do not look like the parents or grandparents at all. So they call these recombinant offsprings. Okay, these are parental offsprings because they do look like parents, either the parent or grandparents. These genotypes, we talked about this. Okay, but these genotypes do not look like the genotype. I'm talking about genotype, not phenotype. The genotype does not look like any of the parents or great or grandparents. Morgan discovered that genes can be linked, but the linkage was incomplete because some recombinant phenotypes were observed. He proposed some processes must occasionally break the physical connections between genes and same chromosomes. That mechanism was crossing over of homologous chromosomes, which we talked about that, crossing over. And genetic map, when you can have genes, you can map them. Of course, it was very easy for Drosophila because he had only 8 chromosomes. But for human, it took until 2003, 2004, I guess, I don't know, somewhere in that neighborhood, they discovered that's the gene mapping. Alterations of chromosomes, number of structures cause some genetic disorder as well. So it's called non-disjunctions. And right here, meiosis 1, meiosis 2, they are non-disjunctions. During meiosis, they are not supposed to separate. They are supposed to separate, but they do not separate. And they call the process, the phenomenon is called non-disjunction. And that's what happens. Let's watch this video maybe. Well, he's going through non-disjunctions in mitosis. But what we are talking about in meiosis during non-disjunctions. It can happen in mitosis too. Aneuploidy, it results from fertilization of gametes in which non-disjunctions occurs. Offsprings with this condition have an abnormal numbers of particular chromosomes, and a monosomic zygote has only 1 copy of a particular chromosomes, and then trisomic has 1 extra, has 3 copies. So this would be 2n minus 1, this would be 2n plus 1. And you all know what 2n means, a complete set of chromosomes. Trisomic is a zygote that has 3 copies of a particular chromosomes. Okay, polyploidy, a condition which organ usually happens in plants, triploidy and tetraploidy. What happens in some plants, meiosis, it means reduction. What happens, they do not undergo meiosis, they undergo mitosis, and then the pollen and the egg are combined. They have full chromosomes, 46 chromosomes, let's say in case of human. Sperm has 46 and egg has 46. and then the offsprings would have 92 chromosomes. Okay, and pretty soon, a few generations later, the cells are full of chromosomes, and that cannot happen. So you must reduce the number of chromosomes if you want to have sexual reproduction. As I said, common in plants and but not in animals. Polyploidy are more animals in appearance. Okay, there are 4 phenomena: A, B, C, D, and each one of them are responsible for a disease. That's what happened with the chromosomes. A deletion, it means a piece of chromosome is deleted. Okay, a piece of chromosome is being deleted. Duplication, when a piece of chromosome is duplicated, easy to remember, right here is duplicated. And then inversion, almost like it's different than crossing over. When this one goes here and that one goes here, that's called inversion. Translocation, I hope there are some pictures. Yes, perfect. So right here, you have A, B, C, D, E, F, G, H. If D is deleted, then you have A, B, C, D, E, F, G, H. That's deletion. Duplication, A, B, C, D, E, F, G, H, A, B, C, B, C. They've been duplicated. Okay, during the S phase of the mitosis or meiosis. And then inversion, if you have, you know, A, B, C, D, E, then it's A, D, C, B. It makes a difference later on when we talk about it. It will make a big difference. Translocation, it means A and B from this chromosome went to that chromosome, to this chromosome, and M and O came from that one to this. That's called translocation. Down syndrome is an aneuploid condition that results in 3 copies of chromosome 21. So it's a trisomy. So it's 2n plus 1. And then in effects, out of the 700 children born in the United States, frequency of Down syndrome increase with age of the mother, of course, in correlation with this one. Okay, sex chromosomes non-destructions produce the triple X females are healthy with no unusual physicals. Klinefelter syndrome, double X with Y, syndrome chromosomes in male producing individuals, and monosomy of X called Turner syndrome, as usually they refer to as a Turner syndrome. It's X0. So it's a usually female that does not have the other X chromosomes. And there are signs and symptoms along with all of these. Your textbook did not get into it. The syndrome cry-do-chat, which is a cry of the cat, results from the specific deletion of chromosome number 5. The baby cries, when the baby's crying, it sounds like a cat. Okay, they call it, that's what they call it, cry of the cat. And a child born with syndrome severely intellectually disabled, so on and so forth. Certain cancers, including chronic myelogenous leukemia caused by translocation of chromosomes. This is translocation between number 9 and 22. Inheritance of organelles, genes, extranuclear genes, and cytoplasmic genes are found in organelles and cytoplasm, mitochondrial chloroplast, and other plants. Plastids carry small circular DNA, is called plasmid. Okay, extracellular genes are inherited maternally. So since our chromosomes are sperm, when the sperm enters the egg, it loses the neck. So there are no mitochondria from male participating in inheritance. It's all female mitochondria. So I do not have any of my mitochondria from my dad. All of my mitochondria is from my mom. And that is true about all of you guys. Everybody's mitochondria is from their mom. And that is, they can detect some of the historic events and so on and so forth. That's a different. First evidence of extracellular genes came from studies of inheritance of yellow. Okay, some defects in mitochondrial genes prevents from making enough ATP results in muscular and nervous system. For example, mitochondrial myopathy and Leber's hereditary optics. Okay, as I said, toward the end is a little bit of diseases. Make sure you know some of them. Okay, well, most of them. All right, guys, I'm done. As I said, it's a short chapter.